Hello guys, how are you? So today we are going to start with a new topic, and that is mitosis. So the topic for today that we are going to cover is mitosis. I hope you are listening to my videos and you're sharing it with your friends, because I think that's a very good way, always to share the knowledge among your friends, among your peers. And so let's start the today's topic, that is mitosis. Mitosis we are learning from very small classes, like I guess it starts from 7th or 8th class, and it's an important topic, easy topic, relatively easy topic, and we'll see all the stages and we'll try to draw the diagrams and understand it and just cover one topic. I hope you are referring to some books even after seeing my video or before seeing my video, because that's important, and you're doing questions. So you should read book. Okay, you should make notes, and you should make diagrams. That's the only way, or one of the very important ways in which you can study biology, zoology and botany both, and you are doing lots of questions. So just do all these 4 things and you will, whatever you want, wherever you want, in whichever college you want, you will easily get through it. See, it's not difficult. It's not difficult. It's just too much. So you have to daily sit and study. So sit and study. Guest, target, chachi, tai, tau, mama, mummy, anyone coming, no. They will understand that you're studying. Okay. So, chalo bhai, gyan ho gaya. Ab thoda padhai kar lete hain. So as I said, today we are going to study mitosis. And what is mitosis? If I just ask you, no definition, what is mitosis? What is happening is my mitosis? Can you just say, what is mitosis? So basically mitosis is what? There's a cell and you're dividing that cell into 2 cells, right? Basically what you're doing when you're saying cell into 2 cells, you're replicating, right? You are replicating. And what is this used for? If it's replicating, it's used for cell growth, right? And what is cell? Chalo, before getting into things that cell into 2 cells, replicating, what is getting replicated and how it is getting replicated, where it is getting replicated, we'll do all these questions just in another 2-3 minutes. Before getting into much detail of mitosis, first let us see if we remember what is cell. So we all know cell is what? The chapter itself says the cell is the basic unit of all living things, or cell is the basic unit of life, right? So basically, so technically there should be 3 things in eukaryotes. We are talking about eukaryotes and specifically human cells. Technically, there should be 3 basic things in eukaryotes to call it a cell. And what are these 3 basic things? If we see, they are cell membrane, right? Cell membrane, there should be a phospholipid, remember, a phospholipid bilayer. There should be a cell membrane. What else? There should be a nucleus with chromatin. We'll see what is chromatin also here. So basically what is chromatin? Chromatin is DNA that's, wrapped, DNA that is wrapped around different types of histone proteins. Okay. So we'll do DNA also in one lecture in great detail. And there should be cytoplasm, okay, in which all these things are present. So basically, this is what is a cell. A cell membrane, there should be 3 things to call it a cell in eukaryote. Cell membrane, nucleus and cytoplasm. Chalo, ye baat to ho gayi. Now let us see something else. What do we, when we say we want to replicate DNA, we want to replicate DNA, right? In mitosis, we always say we want to replicate DNA. What do we actually mean by it? We mean wants to synthesize that the double-stranded DNA. We'll do each and everything in detail. Slowly we are moving forward. चलो ये हो गया that cell को replicate करना है DNA. It has to synthesize double-stranded DNA. This is mitosis. This is what? This is the goal of mitosis. Goal of cell in mitosis, right? ठीक है, उसके लिए what will the cell do? The cell first will go into, there will be a first phase. And what will be that first phase known as? That first phase will be known as G1 phase or gap 1 phase, right? Oh, no need for this bracket, or gap 1 phase. So the first phase is G1 phase or gap 1 phase. Now, what is the cell going to do here? So the question that we should ask whenever we are reading science, we should ask questions to ourselves. So what is the cell going to do here? The cell is making, or it's getting itself ready for mitosis. So what is the cell going to do? The cell is going to get ready for mitosis, right? So basically उसको क्या करना होगा? अगर उसको ready होना है तो उसको क्या करना होगा? As we know, there are how many chromosomes? There are 46 chromosomes, right? 23 maternal and 23 paternal, right? तो एक ये सेल है 2n diploid, इसको basically क्या करना है? Replicate करना है with the same genetic material, same number of organelles. Replicate मतलब बिल्कुल copy कर देना, तो everything should be same. तो इसको ये करना है. तो basically क्या करना है? Replication में, during replication, the genetic material remains same, right? Genetic material remains same, right? That is happening in mitosis. Ab, in the cell, there are other organelles. In the cell, there are what? There are ribosomes and there are mitochondria. Kids, write with me, or else in the end, there'll be too much to know and you will have no notes. So in the cell, there are many things like ribosomes, mitochondria, and other organelles which we know about, right? So what the cell needs to do? The cell needs to have more organelles, right? The cell needs to make all these organelles more, right? Because it is replicating. अब एक है, उसको दो में होना है, so obviously whatever material is there, it should increase it, then only it will be divided equally, right? So equal distribution of all the organelles होना है. तो उसके लिए सेल को क्या करना है? It has to make more organelles. तो और न्यूक्लियस में क्या होता है? Let's see. Inside nucleus, there is what? There is DNA, and DNA is the genetic material. So the cell should make what? The cell should make lots of enzymes and proteins for DNA replication. So basically what is it doing? It is getting ready for mitosis. Okay, so in G1 phase, what happens? The cells get ready for mitosis. Now there's one very interesting thing I'll tell you. Most of the cells, most of our cells exist in G1 phase, right? So some might be in like in 8 hours. कुछ cells होंगे जो खाली 8 hours के लिए G1 में होंगे, और कुछ होंगे जो years and years तक they will be in the G1 phase. So कुछ होंगे, some cells होंगे जो 8 hour में होंगे, and some cells होंगे जो years and years तक G1 phase में रहेंगे. So let us see what are we talking about is some cells. Which cells are we talking about? These some cells, which cells are we talking about? I told you, whenever we are studying science, questions should come in our mind. So there is some kind of cells which is known as proliferative cells, right? Or labile cells. We know from this word proliferative cells, labile cells. What do you understand by proliferative? या proliferation, बार बार ज़्यादा बढ़ना. So इसमें labile cells are what cells? These are those cells that are all the time going to cell cycle, right? ऐसी कौन सी सेल हमें बार बार ग्रोथ की और बार बार रिप्रोडक्शन की सेल को जरूरत है? Imagine we are constantly shedding what? We are constantly shedding skin cells. So epidermis में कौन सी? Stratified squamous tissue of epidermis, right? These are proliferative cells or labile cells. Constantly we are shedding, constantly there is a need to make, right? Urinary tract, right? GI tract, eating food and all the GI in detail. What else? What else can you think? Then there can be hematopoietic stem cells. What are these cells? We have to constantly make what? Constantly make WBCs, RBC, platelets, right? And these are also proliferative cells. Now the second type of cells that we are going to do, this is just a little off that we are going from the mitosis, but it's important, that's why you should know it. We are still on the G1 phase, and in the G1 phase I was telling you that most of the cells exist in G1 phase. So we thought that which cells, when we say most of the cells, we should know what are these different types of cells. So now the other type of cells are stable cells. What are stable cells? These cells retain this capacity, but do not continuously replicate. वो कर सकती हैं, but वो नहीं कर रहे. They can replicate, but they are not replicating. And when do they replicate? They do this when there is a strong stimulus to do so, right? When will they replicate? When there is a strong stimulus to do so. What can be these type of cells? Think. I'll give you an example. We have done this in GI when we are doing. So these are the hepatocytes of liver, and they are like the epithelium of kidneys, loop of Henle, PCT, right? They are stable cells. Then alveolar cells of lung, right? Alveolar cells of lungs. So ये क्या हो गए? ये हो गए stable cells that have retained that have the capacity, but they are not constantly replicating. Now there is also a third type of cells, and these are known as amitotic or static or permanent cells, right? From the name itself, we know that what amitotic, static or permanent cells. इसमें क्या हो गया? They these cells do not reproduce after birth. एक बार हो गए तो बस deteriorate ही होंगे, बनेंगे नहीं. ये कौन से हो गए? सबसे important, we all know this, neurons and the cardiac muscles. And what? One more example, skeletal muscle cell. So they are what? They are permanent or amitotic or static cells. Chalo, ye, we were doing this, the G phase, G1 phase, we had done it. We just stopped and we began to do these three types of cells: proliferative cells, and then stable cells, and then amitotic or permanent or static cells. Chalo. Now we have to come to the synthetic phase. Which phase? Now we have come in. Now the cell has come where? The cell has come in the synthetic or S phase, right? So synthetic phase or S phase. What will happen in the synthetic phase? What is the structure of DNA? If I ask you what is the structure of DNA, so you will say what? Ma'am, it is a double-stranded, coiled. It is a double-stranded and it is coiled double helix. This is the structure. So basically, this is the structure of DNA, right? The DNA will also laugh at me. So basically it is what? It is coiled. Now in the synthetic phase, what will happen? It will right to make a new strand. See? Now there is a new strand that is getting formed. So what is happening? There is this unzipping taking place, right? And why is this unzipping taking place? To make a whole new strand. The name itself says synthetic phase. Something is getting synthesized. So this is the synthetic phase. And you understood this, that is this double helix, double-stranded coiled double helix, right? So basically, if you want to write what happens in the synthetic phase, please write in your copy with me. Your notes should be up to date. When you're revising, you should have notes, because every time you cannot just open YouTube and watch, right? When the DNA, it is the phase of the cell cycle when DNA packaged into chromosomes is replicated, right? And why very essential? Because it is essential. It is replicated. It means it has the same genetic makeup, which is a very important part of mitosis, right? So basically DNA replication is taking place. What will happen? If there was 2N, DNA diploid, replicated, it will become 4N. And there are most prominently, I told you in the G1 phase, there were so many enzymes and proteins the cell was getting ready. So basically there are many enzymes, but the most important and the most prominent enzyme is DNA polymerase. And DNA polymerase 1 and DNA polymerase 2. So this is a very prominent enzyme which is helping it in the replication of DNA. And you know, this DNA replication, this DNA polymerase is doing its job very well, very good. It is a very good enzyme. Why? Because it makes no mistakes. And if we ask that, does it ever make a mistake? Supposedly 1 in million or 1 in billion. So, and you know, it is not only replicating, it has also this DNA polymerase, it also has proofreading ability, you know, proofreading ability, matlab dobara check karna. So it checks for mismatched bases, right? So the mismatched bases जो होते हैं, जो गलत होता है, वो उसको remove कर देता है. And generally, the cell in the synthetic phase lasts for 6 hours duration. Generally, जीवन में क्या था 8 hours or some cells years and years, but इसमें क्या है 6 hours generally, right? And there is one very important thing happening. If this is the G1 phase and this is the S phase, so between this, no, there is what? This is the checkpoint. This is a checkpoint. And why a checkpoint is needed? To check, obviously checkpoint ho to check hi hoga na, to check what? If the cell is ready, first thing, and there are no damages in the cell. Because agar damages hoga to wo forward chala jayega. So it will check and then only the cell will go into the mitotic division. See, even the cell is checking. So you should also check when you are studying that you're not daydreaming and you're studying. Right? Now let us do the other phase. After S phase, what? There is this G2 phase. And what is this G? What is the other name that we can give it to? Gap phase. Very easy. G1, S, and now G2. Now, क्या क्या हो गया है हमारे पास सेल में? क्या क्या है हमारे पास ऑलरेडी? क्या क्या है हमारे पास ऑलरेडी? मोर ऑर्गेनल्स हैं, राइट? हमारे पास डीएनए रेप्लिकेशन हो गया है, राइट? इन द सिंथेटिक फेज डैमेजेस चेक हो गए हैं, राइट? What else is needed? Now in the G2 phase, now cells check for cytoplasm, right? Because what will happen, there is this cell and finally it will get squeezed. This cell will get squeezed and it will become 2 cells, right? So basically हमें क्या चाहिए? अगर साइटोप्लाज्म की जो क्वांटिटी है वो इंक्रीज होनी चाहिए, then only when it will get squeezed into 2, what will happen? It should have the same amount of cytoplasm as this cell. So basically क्या हो रहा है यहाँ पे? साइटोप्लाज्म जो है वो इंक्रीज हो रहा है, so that these new cells have same amount of cytoplasm, right? चलो, so basically इसमें क्या हो रहा है? If you want to write what is happening in the G2 phase, in the G2 phase there is increase in cytoplasm. Okay, so basically what we do, we combine this G1 phase and this S phase and this G2 phase and we give it a new name that is called the interphase. Interphase is where the cell is getting ready, the DNA is getting replicated, and there is increase in cytoplasm. So in the G1 phase, the cell is getting ready, the DNA is replicated, right? DNA is replicated, and what else? And in the G2 phase, there is increase in cytoplasm. How's the cell getting ready? The number of organelles are getting increased. Right? So now let's enter into the M phase. What is this M phase? Now we are going to do this M phase or the mitosis or the mitotic phase. And what are the 4 phases? PMAT, I remember it by PMAT. It's prophase, metaphase, Anaphase, and telophase. And then there is another thing, but we don't call it a step. It is cytokinesis, where the cytoplasm gets squeezed into 2 different cells. So now let us first do the first step, that is the prophase, right? So what is prophase all about? Let us see, right? So we are going to do prophase. Okay, so the prophase, what happens? There is condensation of chromatin. Basically, what is happening? There is condensation of chromatin. Because the chromatin exists in what? It exists in loose structures. It is not very condensed or tightly bound. How's the chromatin? Chromatin is generally in the loose form. So if it's the cell, so the chromatin will exist like this. And how we want it? We want it in a condensed form, right? So basically what is happening? Condensation of chromatin. What else will happen? There will be disappearance of nucleolus, right? Nucleolus we all know. And there will be what? There will be, we'll do each of these steps also. I'll explain you. I'm just writing for your reference what is happening in prophase. There will be formation of mitotic spindle, and there will be beginning of nucleoli breakdown, right? So let us see all these things in detail, what is happening in prophase, right? So basically what happens, so first let us see something about condensed chromosome, right? What is this condensed chromosome that we are talking about? So how is the chromosome? Chromosome has a short arm and a long arm, and the ends are known as, these ends are known as telomeres. And this is known as centromere, right? So this is a condensed chromosome, and it consists of two sister chromatids joined at centromere, right? And during mitosis, the spindle fibers will attach it to the centromere. Now, what is this? Is we talked about condensed chromosome. Okay, so and now let us see what we are talking about. This mitotic spindle. So there are centrioles. We all know about them. Centrioles. So these centrioles have what? These centrioles actively monitor the microtubules organization center. What? Microtubules organization center. ये microtubules organization center क्या करता है? ये basically microtubule होता क्या है? पहले तो ये जान ले. ये क्या होता है? ये cytoskeleton form करता है in the eukaryotes, right? And it provides shape and structure to the cell. Shape and structure to eukaryotic cell. ये आप याद रखना, right? तो microtubule. So basically condensation of chromatin हो गया, और अब क्या हो रहा है? Centrioles जो है microtubule organization central को activate कर रहे हैं, और उससे क्या बन रहा है? उससे mitotic spindle बनेगा, right? So अगर ये cell है और ये nuclear membrane है जो अभी है, intact है, condensation of chromatin materials है, condensation of chromosome है, ये centriole है. So basically what is happening? This is activating the microtubules organization center for the formation of mitotic spindle, right? So we all saw इसमें क्या क्या हो रहा है. Now एक point हमने और पढ़ा था, nucleoli breakdown, right? So nucleoli क्या होगा? Nucleoli begins to break down. अब आपको ये बिल्कुल से you should remember, if the nucleoli is getting broken, what will happen? It will result in what? It will result in what? It will result in the discontinuation of ribosome production. And why the cell will never do anything without use? This indicates what? This indicates redirection. Redirection of what? Redirection of cellular energy from general cellular metabolism to where? क्या हो रहा है? हम क्या पढ़ रहे हैं? To cellular division, right? Okay. So prophase में हमने ये देखा क्या क्या हो रहा है. चलो अब एक बात और आपको बता देते हैं, एक और वर्ड के बारे में बता देते हैं. That's called kinetochore, because probably will come across this word. Basically क्या होता है? This is a disc-shaped protein, and you will read about it in higher classes if you do some or maybe in MBBS. So what happens during mitosis? Spindle fibers are attached where? Are attached to centromere via a protein, via kinetochore, right? तो अगर ये क्रोमोसोम है, ये सेंट्रोमियर है, so there is this protein and these are this mitotic fibers, so they are attached to this, right? So all clearly, so they are attached to this kinetochore like this. They are attached to the centromere, जो आपने देखा होगा, the mitotic spindle fibers, right? So they are attached to the centromere via kinetochore. Now let us see what is happening in the metaphase, right? Chalo, now let us see what is happening in metaphase. So metaphase, you can remember this, what happens in metaphase by a very easy thing. M is for metaphase and M is for middle, right? Now we'll see why this middle term is important. So during this metaphase stage of mitosis in eukaryotic cells, and in this stage, what happens? The chromosomes are second most condensed and coiled. I hope you are writing with me, guys, right? You have to remember the stages. And Anaphase mein wo, you can write it in bracket, that in Anaphase, the chromosomes are most condensed stage. So metaphase mein what happens? Chromosomes are the second most coiled and condensed stage. Now what happened? These chromosomes carrying genetic material information, what did you do? They align themselves where? At the equator of the cell, right? So that's why I said M for metaphase and M for middle. So basically क्या हो रहा है? ये chromosome middle of the cell में आ रहे हैं before being separated into two daughter cells, right? So metaphase में basically they are coming in the equator before being separated into two daughter cells. और ये basically 4% of the cell cycle का पूरा time होता है. 4% of the cell cycle होता है. ठीक है? It accounts for approximately 4% of the cell cycle. Basically इसमें क्या हो रहा है? If we see this, 3 cells we draw, and these are the centromeres, right? It's not centromeres, centrioles with their microtubules, right? With their microtubules coming out like this, okay? So basically what is happening, slowly, they are aligning themselves in the equator. They're still not, but they're trying to align themselves with the equator, right? So what are these? These are sister chromatids condensing, and these are microtubules, and they are what? They are these ones are called astral microtubules. right? So now again, what happens? So they have come to the pole, and they have arranged themselves. How they have arranged themselves on the equator, right? And now this nuclear membrane has also dissolved. So what we can see the remains of the nuclear membrane. What are these? These are fragments of nuclear envelope. Okay, and what else? Then these are mitotic spindles, right? Mitotic spindles, and they are attached to the centromere via kinetochore. Kinetochore is a disc-shaped protein, right? So basically, this is what is happening. Oh, I drew it one only, we can remove this, right? So basically, this is what happens. There is this, you can say that there is this imaginary line. We call it as metaphase plate. It is just an imaginary line, and just for our study, we call it a metaphase plate. ऐसी कोई plate नहीं है. It is what? It is an imaginary line which we draw for just for our study sake. So you remember now why metaphase, M for middle. Now let us see the next phase that is very interesting, and that is what? That is Anaphase. Now M से metaphase, M से middle, A से Anaphase और A से away, just for your remembrance. A से away. So basically what is happening in Anaphase now? In the वहाँ पे सारे align हो गए थे chromosome. Now what will happen in Anaphase? First of all, this is just 1% of cell cycle duration, right? So basically what happens, the replicated chromosomes, guys, write with me, it's very important. Replicated chromosomes are split, and daughter chromatids, whenever I'm saying, just not blankly write, move to, whenever you write, your full focus should be on writing. So basically, जो sister chromatic है, they will move where? They will move toward the opposite poles of the cells. और मैंने क्या बताया था chromosomes के बारे में? Chromosomes reach their maximum condensation, right? बताया था मैंने Anaphase में they reach to their maximum condensation. So basically क्या हो रहा है? You draw and see what I am drawing, you are drawing that or not. So basically, if this is the cell, so what will happen in Anaphase? There are many. I'm just drawing some so as to, so that the diagram doesn't get very messy and you're able to understand, right? So what is happening? They are moving towards the poles. The sister chromatids are moving towards the pole. So what happened? These sister chromatids, are moving to poles of cell, right? So this is what is happening in Anaphase. Now let us see what is happening in telophase. That is the last stage before cytokinesis, where the cytoplasm divides. What will happen in telophase? That's first of all, this is the final stage, right? You should know. As chromosomes reach the cell pores, a nuclear envelope is reassembled. So we can say as chromosomes reach to the poles, like they did in Anaphase, ab kya hoga? What will happen? A nuclear envelope is assembled, right? Is reassembled. And where will the nuclear envelope? Around each set of chromatids, because वही तो गए हैं, वही तो है, वही तो जेनेटिकल मेकअप है, उसी के लिए तो इतना काम हो रहा है. क्या गायब हो गया था? न्यूक्लियोलाई गायब हो गया था वो रीअपियर हो जाएगा, और क्या हो जाएगा, जो क्रोमोसोम इतना कंडेंस हो गया था वो क्या हो जाएगा, वो रिकंडेंस हो जाएगा फाइनल स्टेजेस में, और बैक बेयर आ जाएगा, एक्सपैंडेड क्रोमैटिन के फॉर्म में आ जाएगा. So this is what is happening in telophase. Chromatin becomes to become loose again. So अब इसमें क्या हो रहा है, बेसिकली अगर ये सेल है हमारी, ये they call it this thing is known as the cleavage furrow, not furrow, cleavage furrow. And now what is happening, this nuclear membrane is beginning to reappear, right? And this is what, oh, this mistake I did, you should not do. Now see, this is appearing, chromosomes are appearing. What? Haploid or diploid? Diploid. How many chromosomes? 46. I told you when we started this lecture, I particularly told you I'll be telling you about the human cell. And humans have about 46 chromosomes. So basically क्या होता है, यहाँ पे एक टाइप के प्रोटीन होते हैं, आपको जानने की चाहो तो मतलब just I want to tell you, that's why I'm telling you. ये होता है एक मायोसिन प्रोटीन, right? और एक होता है ऐसे actin protein, right? So basically this is what, this is actin protein, and this green one is what, this green one you can know is the myosin protein. And what are these myosin and actin protein? These are contractile proteins, right? So basically ये cell के ये cleavage furrow बनाने में help करते हैं. So these are what, these are contractile proteins. chromosomes हो गए सब. अब ये क्या हो रहा है, reformation of nuclear membrane, right? So you can see there is reformation of nuclear membrane. And अब इसके बाद क्या होगा, what will happen after this, there will be two cells. ये cleavage जो है ये और cleavage furrow deep होता जाएगा, and there will be what, there will be two cells, right? And there will be equal amount of cytoplasm, and there will be diploid, same genetic material, and the chromosomes, the chromatin, what will happen to the chromatin? It will again become to form in its form. So there is what, there is cytoplasm, ribosomes and everything, mitochondria, right? So what is happening, equal amount of chromosomes, right? Equal amount of chromosomes, as you can see, 2n, and what else, same amount of cell organelles, right? Same amount of cell organelles, nuclear membrane comes, nucleoli comes, right? Okay, so this is the end of the telophase stage. And what happens, again the chromatin becomes what, the chromatin lose या recondense form में, back to its recondense form. So this is what happens in telophase. So basically stages. Yeah, and one thing I just forgot to tell you, this cytokinesis. Cytokinesis is what? This is just basically the cytoplasm divides equally among two cells. Remember? Now tell me, in which phase was the cytoplasm? The cell was increasing its cytoplasm, G1, S or G2 phase? The cytoplasm was getting increased in which phase? The cytoplasm was getting increased in the G2 phase, right? I had told you that the major function of G2 phase was what? To increase the cytoplasm. And इसलिए increase हो रहा था जिससे कि वो equally divide हो सके among the two cells. Okay, so I hope guys this mitosis is clear to you, and this today's lecture is over. It was not a very difficult lecture, but you have to know it. Questions can come. So let us just quickly see if you want to, that what have we done today. So we basically started with what is cell, what should be the 3 things that there should be present in the eukaryote so that we can call it cell. So we said there should be cell membrane, nucleus, and cytoplasm. And we said the goal of my cell in mitosis is to replicate DNA, to copy DNA, same kind of genetic material. Then we studied about the G1 or the gap 1 phase, where we said that the major goal is to prepare the cell. Then we saw the in the G1 phase, the cells get ready for mitosis. And then we saw about the different types of cells, labile or proliferative cells. Then we saw about stable cells and we saw their examples. And then we saw the amitotic or static or permanent cells. Then we studied about the synthetic phase, right? And then we saw that there are checkpoints. Then we studied about the G2 phase, where the cell was increasing its cytoplasm. And then we said that G1 and S and G2 together are known as Interphase. Then we studied about prophase. We saw that condensation of chromatin, disappearance of nucleolus, formation of spindle fibers, and beginning of nucleoli breakdown happens in prophase. Then we studied about each and everything that we have mentioned, all these 4 things in detail. Then we studied about microtubules, organization center, and we saw what is kinetochore. Then we saw what is metaphase, M for metaphase, M for middle. So the chromosomes align themselves at the equator of the cell before getting separated. Then we saw about anaphase, that occupies 1% of the cell cycle duration. The chromosomes reach their maximum condensations and the replicate chromosomes and daughter chromatids move to the opposite poles of the cells. Then we saw the telophase, that's the final stage where the chromosome recondenses, becomes loose, and then a nuclear membrane is reassembled, nucleoli appear, and then cytokinesis, where equal amount of cytoplasm is divided. So I hope, guys, it was a fun-filled session for you, an informative session for you. Please like, share, and subscribe, and stay home and stay safe. Bye.